right? Okay, and then directional growth was like the example I gave you earlier, where the yeast cell grows toward each other. So this is actually a change in the cytoskeleton that extends the cytoplasm outward and grows that way. So that's an example. They actually go into detail how that works, but unless you want to know, I'm going to go over to here. Okay, so there's 4 aspects to consider when we think about how cells fine-tune the response. So I just went over amplification is one way. Specificity of the response, which we'll look at here in a second. And then there's overall efficiency that can be done by what we call scaffolding proteins. So these are large proteins that sometimes bind to several of those transduction pathway molecules to make the amplification faster, because it has them all together instead of spread out through the cell where they have to kind of find each other. I'm not going to really expect you to know these details. I'm going to tell you here in a moment what I want you to know. And then last is how the signal is terminated. Okay. Okay, so signal amplification went over already. Specificity and signaling. Okay, so let's consider this first one here, right? We have a simple receptor. It goes through, it activates relay molecules, and you get a response. Okay, in a different cell, right? So cells vary depending on their receptors, their protein receptors here. It might have the same protein receptor, but on the interior of the cell, the relay molecules might be different, right? So we can get this relay molecule goes to response 1, and here, this might activate 2 different other pathway molecules that would create 2 different responses. So it could actually branch, and one signal could give 2 responses, depending on what happens in the interior, what your pathway molecules are. We also might have it correlated with another signal, right? So our body sends out a lot of different signals, just like you can receive a group text, right, from just several different people. You can receive several messages that might coordinate together, right? So this one might send a signal for response 4, which can be activated only or inhibited by this other signal molecule. Right? So this might actually require activation, or this might inhibit that response. And then lastly, we could have a protein that has the same receptor site for the molecule, but it's a completely different protein, so it's in a different pathway here. Okay, so really what I want you to get out of this is that different cells may have different cell receptors that cause different reactions, responses, right? And they can be coordinated through several different signals and several different protein receptors, okay? So the same signal could end up leading different responses depending on where your cell is, right? Depending on what type of receptors and what type of the molecules they have inside, right? Those protein molecules. Okay, and this was a picture of the scaffolding protein. So instead of having your relay molecules all spread out through the cell, it takes time to find each other. Sometimes they're bound together by an even larger protein called a scaffolding protein, so that when this signal is given, it just kind of, it activates this first one, which can kind of almost immediately, right, activate the others, and then this would cause some other response. Okay, and there's different ways to terminate the response, right? There could be a decrease in the ligand, so your body quits sending out that hormone, or these receptors might be turned off. So I don't expect you to understand this GTP since we didn't go over it, but receptors can be turned off in different ways. Or your second messengers might be broken down immediately. So there's different. Really, I'm not going to ask you anything about terminating the response on the exam, but I just felt like I needed to explain that it's something that varies, and there's different ways of terminating a response. Of course, reality is not so simple. There are a lot of different things that go on in our cell. There's a lot more coordination happening with the cell. You can get into this when you take higher molecular biology, right? Cell biology goes a lot more into this. Okay. So what are the functions of a signal transduction pathway? E, all of the above, right? So this might be something similar to what I asked you on a test. I want you to know that the transduction pathway can amplify effect of a signal molecule. It converts a signal on a cell surface to specific response within the cell, right? It allows different types of cells to respond differently to the same signal molecule. So they vary, they can have different response. Okay, so the last thing we'll look at, real short, is apoptosis, which is programmed cell death. So this is a picture of a healthy white blood cell, and this is one undergoing apoptosis. So what happens is, sometimes our cells are programmed to die, and the DNA gets all chopped up, organelles are broken down, and they start to bleb out. So these little ball-looking things or whatever are called blebs. And then they're digested by special scavenger cells that come in and it basically engulf these different blebs and break it down. So one thing, if we think about this, if everything is being broken down inside the cell, would it be a good idea if this cell lysed and broke it open to the rest of our body? No, why not? Yeah, so there's lots of, like in our lysosomes, we have different enzymes that go in and basically break down everything, right? And it's basically it'd be producing or releasing all of these enzymes that would go in and destroy our healthy cells. Okay, so how this works in a nutshell. So this is based on a nematode, studying a nematode, right? But our cells work in a very similar fashion. There are these proteins inside our cell that will go in and perform apoptosis. They chop up our DNA or organelles, but they're inactive in the cell, and they're kept inactive by another protein that's in our mitochondria membrane. Okay, so these are made pretty much at early on stage of the development of this cell, but they remain inactive due to this other protein. So if there is a signal sent out for this cell to die, it will basically cause this protein in the mitochondria to become inactive. So if this one becomes inactive, these will then not be inhibited anymore. They become active and go in and start chopping everything up. So these are called proteases because they cut, ACE is a cut. So they cut up proteins, and these are nucleases because they cut up, nucleotides. Okay, so we can also have interior signals. So if there is a lot of damage to your DNA or misfolded proteins, this can signal for apoptosis too. Basically, if your DNA is damaged or your proteins aren't functional, your cell's not going to be functional, right? So that cell should be died and recycled for the rest of the body to be used. So apoptosis is actually really important in development with nervous system, immune system, and here we see in morphology. So when our hands are being formed, there's skin tissue basically covering all of our skeleton, right, our digits. And so through normal development, there's a program of apoptosis to kill off all the cells between our fingers, right? So it undergoes apoptosis until you get normal digits with space in between. So if you have a mutation that inhibits apoptosis, you end up with webbed hands and feet because this did not occur during embryology. Okay, so apoptosis can also be involved in some diseases like Alzheimer's. And if you think about it, does anyone know what cancer is? We're actually going to talk about it after.
Cells that refuse to die.
Basically, right? So it's unregulated cell growth. So if there's a mutation that inhibits apoptosis, this could also lead to certain types of cancers. So apoptosis kind of has different applications in our body. Okay, so apoptosis involves all but which of the following? D, lysis of the cell. So we just went over that. Okay, so in a very broad sense, I want you to understand how a signal is transmitted, right? It could be through the cells could be directly contacted, or it could be a release of a hormone or a neurotransmitter, right, by the nerves or the endocrine system. There's ligand binding, signal transduction, and then a cellular response is carried out there. That I expect you to understand those three steps that are important. Really, I want you to know what a phosphorylation cascade is. I want you to understand that these proteins can be turned on and off quickly, simply by adding phosphates or removing them. And understand the importance and mechanism of apoptosis. So, I don't really even necessarily, I'm not gonna ask you anything with a fine tuning signal, because I know this is real last minute. Okay. You guys have questions before Wednesday? All right, you guys have a good evening.